think this is where we left off, and this is going to be the last chapter that we're going to cover for exam 2. So I want to remind you that we did talk about viruses already somewhat when we talked about coronavirus, but as I recall, we also lost power that day. So I'm not sure how much of it you've been through. So we'll go through everything, and if some of it sounds repetitive, then you can just ignore it. Okay, so basic characteristics of a virus. First of all, that they are non-living, and I know that we've been through that before, but they need a cell to be able to make more of themselves, so they are considered non-living. We know that they cause a lot of diseases, and they have a nucleic acid genome, so just like a regular cell, which has a DNA and uses RNA obviously, but has a DNA genome, with viruses, we are actually going to have either RNA or DNA as the genome. And they infect both prokaryotic and eukaryotic cells. So every type of living cell can be infected by a virus. We do have a special name for viruses that infect bacteria, and that's bacteriophages or bacteriophages, or sometimes that's even really shortened to phages or phages. Whichever way you learn it, it doesn't matter to me. And we know that they do rely on living cells to replicate. We're going to look at this a little more in depth, but they're very diverse. So we're going to see differences in their host range, the cells that they have the ability to infect. They're going to be structurally different. They're going to look different depending on the type of virus. They may have can have various shape capsids, and they can have the presence or the absence of a viral envelope, and we'll talk about those. In addition, as I mentioned, they can have a DNA or an RNA genome. And as far as the host range differences, some viruses have a broad host range. So the example here is tobacco mosaic virus, which infects plants, but it can infect over 150 species of plants. Other viruses only infect a single cell type. For example, hepatitis. Hepatitis viruses have a special affinity for the liver cells. Okay, so that particular virus may only be able to infect the liver cell in a particular host. So that would be actually a single cell type. The other example here is influenza, which specifically infects lung cells. And we know from coronavirus and looking at both SARS and SARS-CoV, which is our current coronavirus, our novel coronavirus, that they use a spike on the viral envelope to attach to what's called an ACE2 receptor on the cell surface. ACE2 is part of our renin-angiotensin blood pressure control. So it's present on a number of our cells, but it's present in high number on lung tissue. It's also present in the upper respiratory tract, the colon, and the ileum. The ileum is part of the small intestine. All of those cells are prone to infection by coronavirus. Viruses show a wide range of structural differences, including size and the actual structure of the virus itself. First of all, most viruses are very small. They range from about 20 to 400 nanometers. Remember, nanometer is 10 to the minus 9th meter. And normally, we measure bacteria in micrometers, so it would be 1 micrometer or 1,000 nanometers, so you get an idea of the size. There is actually, in 2014, there was a large virus isolated, and that is actually 1.5 micrometers, so it's in the bacterial range size, and it was unearthed under some permafrost, and is estimated to be 34,000 years old. and they actually revived it. So rather interesting. So it's stored for all those years. And all viruses have a capsid. Okay, that's one thing that's common, is they will have a capsid, and that capsid is composed of protein subunits. And those can be basically helical or polyhedral, either one, although there are some other shapes as well. And then some have a viral envelope, and some have specialized proteins to help facilitate infection, like spikes and knobs. This slide shows some various shapes and sizes of the viruses. So I would really like you to look down here to start in the right-hand corner, and this will give you some perspective in the size of viruses. So here's a red blood cell, and next to the red blood cell is an E. coli bacterium. And that bacteria is basically about 1 micrometer wide and 3 micrometers long. So that gives you an idea here. And next to it is an example of a virus. That's a smallpox virus. And this little goober here that looks kind of funny like a spaceship, that's a bacteriophage. That is a virus that specifically infects E. coli. And then there's a couple other viruses. Poliovirus is a human virus, and this other bacteriophage is a bacterial virus. Down here in the right-hand corner is tobacco mosaic virus, the one that infects a number of plants. So looking at the left-hand side, this is the capsid, and the capsid is in the center in this case. That's a helical capsid. This is a polyhedral capsid, and they can have any number of sides. So some of them will have more sides than this. But that is the capsid will house the genome, whatever that genomic material is. So that'll be inside. And we can see over here on the right-hand side up in the top that we're seeing the genome inside, and we're seeing the capsid. This is actually what's called a naked capsid. It's not going to have an envelope around it versus these that have an envelope, and that envelope is made of the cell membrane when the virus breaks out of the cell.